I will talk today, good morning, first of all. So I will talk about advanced imaging of brain tumors and the three main projects I have been focusing on during these 2 months. I focused on PNETs, which are primitive neuroectodermal tumors, and particularly on MN1 mutated astroblastoma, which is a particular subtype of PNET. And then I focus also on other PNETs, which are the GBMs with PNET features. And then in the second part of the presentation, we will see new techniques for having a faster acquisition of MRIs. So first of all, before starting talking about the PNETs, I would like to talk about the different types of classifications that we have for tumors. So tumors can be classified according to their histology. So what happens is that they take a biopsy of the tumor, and put it under a light microscope, and they see the morphology of the cells. And I can classify tumors according to morphology of their cells. Otherwise, I can classify tumors according to their genetics. So I just sequence the nucleic acids in the genome, and I classify tumors according to this. The third thing is epigenetics, which is quite new. We can classify tumors according to the modifications to their gene, to the structures of their DNA. So some groups like a methyl group, which is attached to the structure of the DNA, if we can check all these methyl groups and other groups, we can understand the epigenetic profile of tumor, and we can classify it according to this epigenetic profile. Now, I said this because before 2016, PNETs, which are the main topic of our discussion today, which are primitive neuroectodermal tumors, they were classified according to histology, so according to the morphology of their cells. PNETs are very aggressive tumors. They are fully differentiated and highly aggressive embryonal tumors, which occur usually in young children, but also in adolescents and young adults they are possible. And they were indeed classified according to the morphology of their cells under a light microscope. Then in 2016, a group of authors basically proposed a new classification, which was based on DNA methylation profile and also on genetic characterization. So what happened is that basically they took the DNA of these patients and they analyzed their epigenetic and molecular profile, and they were able to better characterize some of the already known CNS tumor entities, but also to find 4 new CNS tumor entities. And one of these new tumor entities is the MN1 mutated astroblastoma, which is a tumor I was focusing on in these months, basically. But before going to the MN1 mutated astroblastoma, I would like to give like a brief introduction of the physics of MRI, like something very, very quick, actually. So we know that the body is full of protons, we are full of protons, and the magnetic resonance imaging, which is just a large magnetic field, basically utilizes these protons to measure the tissue, to see the tissue morphology in our body. So we know that protons, the spin of the protons create a magnetic field, magnetic field. So when I put these protons inside the bigger magnetic field, what happens is that these protons will realign according to this magnetic field. Now, if I want radio frequency poles, what happens is that these protons basically realign by 90 to 180 degrees with the big magnetic field. If I remove these radio frequency poles, these protons will go back to their initial realignment, and they will release some energy while moving to the initial realignment. This energy and time it takes for tissues to release this energy is measured by a machine, which understands the morphology of tissues thanks to this release of energy from that. So this is a very basic physics to understand the different sequences that we're going to see in just a few seconds. So we're going to talk about this MN1 mutated astroblastoma, which presented in a healthy 9-year girl. It is a very particular tumor. It is very rare, and we're going to see what are the peculiarities of this case report and of the case of this young girl, which is very, very peculiar. But before going to the different sequences of MRI, so to the images of tumors on MRI, I would like to explain what are the different sequences of MRI. We have many sequences, it's not just one image. We have many different images. The first one is the T1 weighted image, which is usually utilized to see anatomic details of the brain. And if we administer contrast to patients, so if we inject gadolinium, which is a contrast agent, in the veins of patients, basically it goes all around the body. It can also reach the brain. When it reaches the brain, it can accumulate in the brain if there are some damages. So if there is a vascular change or there is a disruption of the blood-brain barrier, which is a barrier which is present between vessels and the parenchyma of the brain, so the matter of the brain, what happens is that gadolinium gets into the brain and accumulates into the brain. And we can see it as a bright signal on T1. So this is the T1 of our patient. We can see this lesion. So this is the M1 mutated astroblastoma we were talking about. It's a very huge lesion with also very aggressive features inside. We can see here, we can see that there is necrosis, which is a sign actually of aggressiveness, and also very huge mass, which is heterogeneous. Now, what is the point? Why is this case so peculiar? Because while we're seeing a very aggressive lesion and a very big lesion on MRI, what we can see also is that we have very smooth borders around and no infiltration of the tumor. And also the mass is very superficial. So basically this patient is doing very well. Although the prognosis of this patient was expected to be a very bad prognosis, actually this patient after 3 years and 6 months is still alive without recurrence of the tumor. And this occurs because the smooth borders of the lesion allow gross total resection of the tumor, so no tumor cells remain in the brain of the patient, and this accounts for no recurrence. This is so interesting because if a clinician sees this lesion, they may think that this is an aggressive lesion. But we are saying that thanks to the smooth borders, we can remove the lesion, and so we can prevent maybe a lot of additional treatments which are very severe, because if I know that the prognosis is good, I can avoid some chemotherapy, some radiotherapy, which can decrease basically the quality of life of the patient. So this is very good. And now, if you want to see also the other sequences, we can see that also gadolinium pathway is really heterogeneous with some neovascularity in the posterior portion of the lesion, so some aggressive features as well. Then, if we want to study this lesion also in T1 is another sequence which relies to see mainly CSF, cerebrospinal fluids, and also to see a lot of lesions in the brain. The problem of T2 is that it cannot distinguish lesions from CSF. So if I have a lesion which is very close to the ventricles, where CSF is, I'm not able to study that lesion because it appears maybe bright or there is too much brightness from the CSF. So what I can do is using FLAIR, which is fluid attenuated inversion recovery, which is just like a T2, but with attenuation of fluid. So basically, instead of appearing very bright as here, as in the other sequence, here the fluid appears dark. So basically, I can see the lesion around the ventricles. I can see also edema and some grade 1 matter differentiation. So it's very good sequence as well. This is the FLAIR of our patient, and we can see that there is just a little edema here. Now, what is important here is that there is no infiltration of the tumor. So again, smooth capsule, no infiltration of the tumor, so burst of the resection is possible. Another sequence is the ADC, the diffusion of the DWI ADC, which is a sequence which can be utilized to study basically diffusion of water molecules. Water molecules usually can freely move among cells, basically. But if there is a malignant transformation as here, we can see that actually we have a lot of densely packed cells and a lot of high nuclear to cytoplasm ratio. So what happens is that basically water molecules cannot move anymore because there are too many cells around. So these basically restricted diffusion of water molecules can be seen on another sequence, which is this one. We can see here that this region of very dark signal means that the water molecules are not moving. So there are a lot of cells in that region. And this is a sign of aggressiveness. So again, I see this lesion, I think that this is an aggressive lesion, but actually there are smooth borders. This is the last sequence we are going to see, which shows again a very huge mass with which is disrupting the fibers around. But we can see again that there is no infiltration. This is a very beautiful image because it shows the direction of the fibers in the brain. According to the color of the fibers, we can understand the direction of the fibers. For example, the green color means that the fibers are oriented on the y-axis, the red one on an x-axis, and the blue one on a z-axis. So we can easily understand actually the position of the fibers just by looking at the color of the lesion. And we can see that right after surgery, but also 3 years and 6 months after diagnosis, we have a patient without tumor recurrence and with just a small lesion, post-treatment lesion here, thanks to the gross total resection of the tumor. So what are the conclusions? That aggressive features do not necessarily correlate with aggressive behavior as soon as gross total resection is achieved, and that histologic and imaging features combined with molecular and epigenetic profiles would allow more clear classification and prognosis, so favoring individualized therapy. Now, not all the PNETs are the same, of course. We have seen one type of PNET, which is the M1 mutated astroblastoma, but we have also many other PNETs, like, for example, the glioblastoma multiforme with PNET features. Now, this one is not a glioblastoma with PNET feature. This is a glioblastoma without PNET features. I put this one because I want to show the morphological difference between a glioblastoma without PNET feature and a glioblastoma with PNET features. Because what we found actually is that all the glioblastoma with peanut features, they are similar to each other. So basically we have always the very same morphology on imaging, almost always the very same morphology on imaging. And it is different from a glioblastoma multiforme without peanut feature, which is this one, which is a very aggressive tumor, which is infiltrating, really heterogeneous. So now we're going to see 5 cases of GBMs, glioblastoma multiforme with peanut feature. What we can see here is that this lesion is very homogeneous actually on T1. It's very dark, this lesion, of course, right? So it's very dark on, it's very hyperintense on T1, hyperintense on T2, very round and homogeneous. So it may look a benign lesion, or actually it looks more like a brain metastasis. But anyway, it's very homogeneous in appearance. But what we can see here is that there is some infiltration. So basically, even if the lesion is very small, there is infiltration. So this lesion is much worse than the previous one, just because even if it is small, there is infiltration in the brain parenchyma. So there is no possibility of removing the entire lesion. Indeed, on follow-up, we saw that there is some recurrence here, because there was no possibility, even if we resected maybe more parenchyma of the brain, there is still recurrence because there was no possibility of avoiding infiltration of the tumor. This is the second case. We can see again that this GBM with peanut feature looks very much like the previous GBM, but it doesn't look like GBM without peanut feature that we saw at the beginning of the presentation. It is big, but it's, I mean, the morphology is very similar, and there is again infiltration here. So also in this case, we have recurrence of the tumor here. This is post-treatment changes, but we still have recurrence of the tumor because we couldn't basically remove the entire lesion. This is case 3, same thing. So the lesion is very similar in all the sequences, but there is infiltration in the surrounding matter of the brain. And indeed, we have on follow-up, again, the lesion is still here, so no possibility of removing the entire lesion in this patient too. And this is case 4. It looks different, but actually it's not that different because we can see like this hyperintense on T1, hyperintense on T2, then there is rim enhancement here, and also the lesions are round. So the morphology is similar to the previous ones. I don't even have another MRI for this patient because, I mean, the situation is too sad. There is nothing to do for this patient actually. Now, the only particular case of GBM with peanut feature was this one. This one is different from the other ones. We can see that this lesion is, I mean, it seems like the MN1 mutated astroblastoma, right? And we can see that inside we have a lot of aggressive features. We have necrosis, we may have some hypercellularity on ADC here, but we have, again, smooth borders and a beautiful capsule. So we know right now that we can predict the prognosis of this patient, and we can see that we expect these patients to do very well on prognosis, right? Because there is capsule around the brain. Indeed, after 16 years, this patient is still alive without recurrence of the tumor. And this is very interesting because this may look aggressive, while instead it is not, as we said. Now, for the last few minutes, I would like to focus on a new technique of MRI. This is the last project that followed. We have basically, right now, in order to get sequences of MRI, we need to get a lot of different acquisitions. So basically, for each single sequence, we need one acquisition. With this instead technique, we can basically have just one single acquisition to have many different sequences. So in 6 minutes, I can get the quantitative features of the tissue, then I put them in an algorithm, and thanks to this algorithm, basically, thanks to this software, actually, I can have all the different sequences that I need in a very short time. So what we did was trying to understand whether this very good tool, which can allow MRI to be taken in a very short period of time, is as good as the standard sequences that we have on imaging, so on the actual imaging of the brain. And we noticed actually that in the majority of cases, like in all the cases that we studied, SWI stage sequences were better than standard test sequences. In most of the cases, stage T1 sequences were better than standard T1 sequences, but in none of the cases, stage T2 sequences were better than standard T2 sequences. And this is really, really interesting as well, because it means that this is a very good tool that we can use in clinical practice, but it needs a lot of improvement before it can be utilized, of course. But if we can utilize this, I mean, it's a very good improvement because it's faster. It does a lot of things automatically, like calculating volumetrics of the brain or segmenting the brain automatically, and also you can calculate automatically microbleeds. So this is very good. These are just as well, the outside the subthalamic nucleus are just the main, basically the main structures of the brain that we studied in order to understand whether one sequence was better than the other or not. So it's just for that. And so yeah, these are the conclusions, and that's all. Thank you.
Thank you very much. I think your talk was really the proof that we were right in thinking about this instrumentation link, you know, this link between medicine and advanced instrumentation. So it's just a proof that it worked. It's a paradigm which works. So thank you very much. You are taking
a beautiful image at the lake with
okay, good. So questions, comments. Oh, yes, Emma, please.
So you used the first line of the talk. You talked about smoothness of tumors. Is there some way to quantify? How would one teach a computer to recognize the smoothness?
Okay, this is a very good question. Actually, we didn't think about that much, actually, because you mean if we can use like the synthetic MRI on the computer to calculate the smoothness? No, we didn't think about it, but I think this is really interesting, and this may be important. I don't know if we have the informatics tool to do that right now, because we are still trying to understand how the lesion is structured inside, to segment the brain, to calculate the volumetrics. So things which are maybe more advanced right now in terms of informatics and software. But yeah, this would be interesting to do. I don't know right now an answer about it, but thank you.
Good.
Yeah, I also, well, I guess I have two questions. One is sort of broad. If you could tell me a little more about the stage, either the sequences or the reconstruction, and then I guess a specific question about those is, it looked like your analysis was still qualitative. I wonder if are the SWIs and the T2s, are they quantitative images at all, or have you looked at that, or is it you're just looking at whether or not the
okay, yeah, I see your question. Okay, thank you very much for this question. I didn't have time to go in more details about that, but thank you for giving me the chance to do that. So basically, the point is that the quantitative, like I get the quantitative features, the software gets the quantitative features of the lesion. So you measure things which are inside the lesion by applying mathematical tools. And then
is it like a deep learning thing, or is it like
I think it's an AI thing, because we are using this tool in order to improve our artificial intelligence, to apply artificial intelligence. Because if I have just one single protocol, like in this case, I can apply artificial intelligence in a much simpler way than having a lot of different protocols with the different acquisitions. So actually, this software is able also to do some calculation by itself, like the number of microglids and some volumetric analysis, which can show that it's much better also in understanding that. Because, for example, if we don't have this tool, we are able only to calculate manually, basically, the number of microglids. So it is able to do also this type of quantification. Our study is focused on the imaging. So it's focused more on comparing the standard sequences with the stage sequences, and to show also that it is able to do some mathematical analysis and some mathematical calculations by itself, which would, of course, make things much faster. Did I answer your question?
Yeah, that's very helpful.
Okay.
Okay. Time for another? One more? Okay. Very good. Thank you very much.